Hello, everyone. Good afternoon. My name is Pedro Raposo, and I'm a curator at UniProt. In this webinar, myself and Nadia Urakova, another curator at UniProt, are going to talk about automatic annotation systems in our database. We are going to discuss how UniProt uses these systems to add annotation to a large number of uncharacterized entries. So we will start with a short introduction on UniProt, and then we will look into the annotation systems in depth. And afterwards, we will finish on how UniProt made it possible for you to annotate your own protein datasets using the same automatic annotation systems. So to start with, UniProt is the world's leading high-quality and freely accessible resource of protein sequence and functional annotation. It has reliable data as we have teams of expert curators reviewing literature in order to describe what the protein does. And UniProt is a massive resource as it has currently 250 million proteins that cover more than 1 million different taxa. The UniProt consortium is composed of 3 teams, and these are EMBL-EBI from the UK, the Protein Information Resource, or PIR, from Washington, DC in the US, and SIB, the Swiss Institute of Mathematics in Switzerland. And together we coordinate our effort to provide you with the many services that you can find on our website, as well as a vast knowledge in protein information. So just as a brief introduction on the different UniProt resources, we start by importing information from external genomic sources, such as ENA and GenBank, for example. And then the information provided goes into UniPark, our archive resource for protein sequences. And then we transform those sequences into protein accessions in the UniProt knowledge base, or simply UniProt KB. We provide datasets for clustered proteins based on sequence similarity in UniRef. And finally, we group proteins by their genome assemblies, creating proteomes. But today we are going to focus on the UniProt KB. So the UniProt knowledge base is composed of 2 different datasets, SwissProt and Tremble. SwissProt contains records that are reviewed, which means that the curator read the literature with experimental evidence on this protein and added those annotations to the record. We can identify these entries by the golden icon with the star. And at the moment, SwissProt has over 500,000 records. And on the other hand, we have Tremble, which is a dataset that contains unreviewed records. And at the moment, we have 250 million proteins. We can identify this by the gray icon. So the types of information we provide in UniProt are diverse. For example, some are the protein name, the function description, the taxonomy of the organism in which the protein came from, the subcellular location of the protein, the 3D structure, domains and motifs, the canonical and isoform sequences, cross-references to other databases, etc. It would be great to have curation for all the proteins. However, some proteins do not have experimental data. So how can we characterize those in that situation? Moreover, how can we curate the exponentially imported number of proteins coming into UniPark? There's a rapid increase due to advancements in sequencing technology, which allows scientists to sequence genomes faster and more reliably. So taking this into account, manual curation is not enough to characterize all proteins. And to address this issue, we need to use automatic annotation systems to broadly and reliably characterize proteins. So initially, when UniProt creates a protein, it is unreviewed, and these proteins reside in Tremble. If this new protein has experimental data available, then the curators are going to pick this up and review it. With this, the reviewed record now goes into SwissProt. So automatic annotation systems go on the opposite direction of manual curation. They look into the SwissProt space and they try to infer information and propagate that into Tremble. So as a summary, the goal of automatic annotation is to provide functional information to proteins that cannot be manually reviewed. And these systems, as we are going to see in the next slide, they are reliable and they are traceable. We can start by thinking UniProt EBI as a space where many proteins exist. This covers a wide variety of types of proteins that come from many different organisms. So firstly, what we are going to do is actually grouping these proteins based on similarity. Then what we observed inside each group is that we are going to have a mix of proteins that have been reviewed, colored by orange, and proteins that are still unreviewed, colored by blue. So we can think of this a bit more abstractly. We can say that each group is composed of a SwissProt space and a Tremble space. After confirming that the group has homogeneous proteins, and by that I mean that all proteins from the same group have the same annotation. After we verify this, we can propagate information from the SwissProt space into the Tremble space. And this is because, again, all proteins from the same group are very similar to each other. So each time we infer information this way, we say that we have a rule for this propagation. Finally, we do not consider groups that do not have any experimental evidences because we cannot infer any information in these cases. So this is another way of visualizing what we just saw. In the first example here, in group 1, we have grouped these proteins together as they are very similar. If we know the function of the orange proteins, the ones that have been reviewed, then we can say that the blue proteins that the unreviewed also have the same information. So we can infer the information from SwissProt to Tremble. Now, the types of annotation we can propagate really depends on what is originally curated in the SwissProt proteins. So, for example, in the first group, we are going to propagate the protein name and the EC number. But in the second group, we are going to propagate the function comment in the subcell location. This is an example of a rule in UniProt. We see it is divided into two parts. The left part are the conditions and the right part are the annotations. So the conditions describe how we grouped these proteins together, what they have in common. In the second case, and in this case, we group proteins that have this PFEM signature described here, and they come originally from eukaryotic organisms. We're going to see later in the next slides what signatures are more specifically. And at the end, the Tremble proteins with these two conditions will automatically get the annotations described in the right panel. So they are going to get a protein name, subcell location, etc. Just to show you how this looks like in the end, we have here a Tremble entry, and you can see this protein comes from human, and it has the function command required for normal Golgi function. You can see that this comment has been propagated by our automatic annotation systems by looking at the tag attached to it. You can even click on this tag to see the types of systems and the specific rules that they use to propagate this annotation. This is how we make our annotations traceable. You can see this was provided by both UniRule and ARBA systems. We are going to look into more detail on this, but basically UniRule is a system where curators curate and maintain the rules, whereas ARBA is a fully automatic annotation system. Both work in different ways, but they are coordinated to work together. So when it comes to UniRules, these can be created by EBI, PIR, or SIB, our three teams, and this is why we can see different IDs associated with it. So there are three examples at the bottom of this page, but each rule also has a universal UniRule ID, starting with the letters UR as UniRule. So historically, Each of these teams had their own systems to propagate information by manually creating rules, and afterwards, all these have been brought together under one name, which is the UniRule. And you can search our UniRule rules in the search bar in our homepage. Now that we covered what a rule is, I'm going to explain a bit more detail how these are created and maintained. We start by looking at the SwissProt into our SwissProt space, and we can see that this record has been reviewed because we can see the golden icon at the top. If you go into the family and domain sections, then you can see the PFAM signature that we used before as a condition. So what are signatures and why are we using them in rules? Well, to simply put it, a signature or a hidden Markov is a hidden Markov model, or HMM, which is a probabilistic model that describes functionally a region of a protein. An HMM can be small in length and can describe the domain of a protein, or large enough to describe the function of a protein as a whole. And the purpose of using these signatures is to group proteins together so we can infer information using rules from the SwissProt space into the Tremble space. These signatures are found in Interpro database. So Interpro is a conglomerate of information coming from 15 different databases with data on the functional classification of signatures. There are a number of signature databases that try to infer protein function in different ways by using different methodologies, but at the end, UniProt puts all this together into one single database, and we get the signatures from Interpro. This is an example on how Interpro displays signatures for a protein. Each color bar represents a different signature, and the top horizontal gray scale represents the protein with 800 amino acids in this case. And we can see on the right the signatures of this protein, and we can see exactly the PFAM signature that we used in the previous rule. So let's see how we create a rule using the PFAM signature we just looked at. So firstly, we need to define our space, our Tremble and SwissProt space. So in order to do that, we can go into, this is our homepage. You can go into the advanced search, and if you click on it, you can search proteins by different attributes. We can actually choose the attributes that we want. So instead of a gene name, let's just select PFAM. So we just write PFAM, and we can add our PFAM ID to get all the proteins with this signature. So this is going to be our group. And also we can do the same thing with other characteristics. So we can look into proteins that belong to a eukaryote. After this, you can click the search, and this is our result page. We found that the signature is present in 3,239 proteins, where 35 of them have been reviewed and 3,000 have not been reviewed. So this is going to be our SwissProt space and our Tremble space. We can infer information if all of these 35 SwissProt have a consistent annotation. We can infer that information from SwissProt to Tremble. Now, this is an example of 3 out of 35 different SwissProt proteins. And you can find something interesting looking at the annotations. And in this table, each row represents a protein, and each column represents an annotation. And all these 3 proteins have the same name, the same similarity comment, same subcell location. So the annotation overall seems consistent with each other. And because the annotation is consistent, we are confident that the annotation from the reviewed proteins can be propagated accurately to the unreviewed proteins that have the same signature. So much so that we have a 95% confidence on all of our applied rules. And with this, we are able to create a rule to propagate information to those 3,000 Tremble proteins. But before we do that, we notice that the function comment is quite different for fungi and non-fungi organisms. All these nuances are taken into account when creating rules. So this is the same rule we've seen before, but now we have taken into account the different functions that we just saw. As we've seen before, that would depend on the taxonomy of the protein where it originally came from. So if the organism is a fungus, then it will get the first comment. And if it's not, it's going to get the second comment to describe a different kind of activity. And this fine-tuning, when creating the rules, allow us to propagate information more precisely. So these are the types of annotations we can add to the rule. This ranges from function, pathway, genealogy terms, EC numbers, etc. And all these enable the user to have a clear picture, not only what the protein does, but also all the context around it. We are able to add information from the sequences as special conditions as well. There are two different types of annotation being added here. First, bioinformatics software for transmembrane regions and signal peptide predictions are run on each of these sequences. And this particular protein has some evidence on transmembrane regions, so that would be part of the annotation in this rule. And secondly, there is a more closely curated methodology of adding sequence information. This is particularly something that is done in HAMAP rules. Here, the protein is aligned with other template proteins, and if there are certain similarities in the sequence, then it is considered appropriate to add that certain annotation. So in this case, for example, if we align the Tremble protein to the template, and let's say a lysine represented by the letter K was found as an amino acid in the 90th position, then we would add the corresponding annotation binding site coenzyme A described here. The system I described so far, the UniRule, has rules which have been curated manually by curators. We have another automatic annotation system, but this one is fully automated. It is important to note that they do not operate in the same way, and they complement each other to provide as much information as we can to the user. So this system is called the Annotation Rule-Based Annotator, or ARBA for short. It also uses the manually annotated set SwissProt as a training data set. And firstly, it loads this data, and then it filters in order to create a highly reliable data set for training. In the next step, then it creates the rules, it selects the most reliable ones, and it aggregates them in order to remove redundancy between each other. And it also performs cross-validation in order to ensure the quality through the statistical analysis. And then it outputs these as a report. And in the end, it propagates predictions that go into Tremble. So ARBA is a multiple class prediction system, which means it generates a model for all the annotations we have previously mentioned. ARBA collects an exhaustive knowledge that is available in all data and selects only the rules that satisfy the statistical confidence criteria. These rules are then subsequently filtered down to sets of representative rules. So this means that we are filtering out rules to predict the same subsets of proteins that other rules already predict. So there's no overlap between them. Now that we covered individually how UniRule and ARBA works, let's go through the differences and how these complement each other. ARBA generates one annotation for each rule, whilst UniRule contains many annotations for each rule. Another difference is that one protein can have multiple annotations coming from multiple ARBA rules, but it can only be touched by one UniRule rule. And at the moment, we have 25,000 ARBA and only 7,500 UniRules. Again, because the difference is ARBA is completely automated, and UniRule has been created and maintained manually by curators. So in this UniRule rule that we previously shown, you can see that many annotations are contained in it, but in ARBA, we just have one annotation per rule. And additionally, UniRule and ARBA provide sequence feature annotations. They annotate certain regions of the protein sequence. For example, they are flagging where a certain domain is predicted to have domain signatures from PFEM, SMART, and ProCite. Also, we are adding the result of sequence analysis methods, or SAMs, and these proteins, and these predict disordered regions on the protein sequence. They predict transmembrane regions, signal peptides, or coiled cold regions. So these sequence annotations cover around half of Trimble, as we see here. Another system we recently introduced is a protein natural language model, or ProtNLM, developed by Google. We have a collaboration with Google on the validation of this tool. And what ProtNLM does is that it uses a transformer model, which is trained on the protein sequence alone to predict its name for uncharacterized proteins. At the moment, it uses an ensemble of 7 different models to predict the name. 3 are going to analyze the sequence, the protein sequence. 3 are going to analyze the organism taxonomy. And an additional one is if we have an AlphaFold structure, a 3D structure. Put it simply, each model provides a score, which is calculated at the end to have the final score and to provide the name. Now, this diagram here represents the number of proteins in UniProt knowledge base. We have 251 million, as we can see, and this is represented by the green, the darker green circle, whilst the red circle represents SwissProt with 570,000 proteins. So overall, UniRule and ARBA together cover 53% more or less of Trimble, which means that for at least for half of our proteins in Trimble, they are annotated by at least one rule from UniRule or ARBA. And if we take into account all of our automatic annotation systems, we can cover 89% of Trimble. So this would be 222 million. So as we can see, our automatic annotation tools are crucial to propagate information and to provide you the latest data that we have. Not only for not only for SwissProt, but also for Trimble. So we know that functional annotation, that the systems are very important and valuable for the scientific community. So we are sharing this knowledge for the of these rules to the users. So in order to share our rules, we have created a file format in which we can represent UniRule and ARBA rules. We have called it the the UniProt rule markup language, and it is based on well-supported and understood standards like XML, and it exposes the logic of the rules explicitly. To allow the users to use these rules, we have created and released as an open source software a rule engine called Unifier that allows the user to use the rules and annotate their own proteins. So as you can see in this example, the UniRule XML exposes the logic of the rule explicitly. For example, on the left side, it shows the combination of several conditions here. In this case, we have a signature condition, a taxonomic condition, and a sequence length condition. And on the right side, you can see the examples of annotation that this rule is providing. The UniRule engine allows the user to use the rules in the UMRL format in order to annotate protein sequences. And the workflow described, it is described in this diagram. So the inputs, you need to have 2 inputs. The first one is the protein sequence, the taxonomic data associated to that protein, but also you need to provide the signature data provided that comes from Interpro. Then the rule engine of Unifier is going to use our rules from UniRule and ARBA to predict the annotations for each protein. So the unifier requires several tools and dependencies to be installed, but you don't need to do all of this manually because we have unifier as a container and that it can be used by Docker, Singularity, or Podman. So the only thing you need to install is one of these three softwares and you're ready to go and run unifier right away. So in order to annotate your protein sequence, the first step is to prepare an input data. So you prepare your first protein sequence data in a multi-FASTA file. The head of each protein needs to be in the UniProt format. It needs to contain at least two types of data, the protein identifier and the taxonomy identifier for each of the sequences. The tax ID can be on whatever level of the taxonomic tree. So it can correspond to a species level or a genus level, etc. However, the more specific your tax ID is, the more specific the annotations we can predict, and the more annotations you may get. So in this slide, it shows the steps that you need to do in order to run unifier in your environment. In this example, we are going to use Docker, but you can use Singularity or Podman in the similar fashion. First, you check that the Docker is installed, and afterwards, you clone your publicly available GitHub repository for unifier, and this repository comes up with a script that runs everything you'll need. The script provides a couple of options, but for most of these options, we have good default values, so you don't need to touch this. All you need to give to the command line is the script. To the script is the input file and the output directive. Those are the only two things. And with this set, you can simply run unifier quickly to give annotations coming from Interpro with our standards to your own protein set. So if you would like to read further information on UniProt, we have released a paper about our database. If you want to read more about UniRule, this is our paper on it. And on ProtNLM developed by Google, we have a preprint available as well. And with this, I thank the whole UniProt team and our funders that support us. Thank you. If you have any questions, Nadia Urakov and I can answer them now.
Thank you, Pedro. I think just while we're waiting to see if there's any more questions, I just wanted to raise a couple of people had asked about the slides for today. And as I mentioned at the start, we'll send out the link to the recording tomorrow. We'll also send out the link to the slides along with that. So you'll be able to come back and have a look through anything that you need to do. And I saw that there was a couple of papers there, so they'll be listed in those slides as well, so you can find those more easily. As we don't have any open questions currently, I saw that Nadia, you answered a couple. Was there anything there that you wanted to raise sort of out loud, as it were, as opposed to just typing?
Yes, so the questions concerning the evidence tags and how you can filter the information and how to understand how the annotations were assigned. So basically, Pedro, would you like to comment on that?
What would be the exact question again? Sorry. Oh, it's in the Q&A. Is that it?
Yes.
Questions and answers. So it's this one. When downloading a protein or proteins, let's say for a proteome, how can I filter the data set for how the annotations are assigned? Yeah, so for our annotations, we have a tag associated with it. So this tag is going to demonstrate you where this information came from. If it comes from manual creation or from an automatic annotation system. It also has some evidence codes. And so it specifically says how this information was manually created. So if it was inferred by homology or not, for example, And when it comes to automatic annotation, we have so we can distinguish if it comes directly from UniRule, ARBA, or, for example, Google ProtNLM. So you can, we have an API that you can use that because we have that information on those proteins. So this is something that you can use our API if you want to filter them. Also, I believe in the advanced search, you can. I think that there's an option on the advanced search where you can you can search if the. Yes, Nadia.
So in advanced search, you can select for experimental evidence. So you can select the type of information that you would like to search, and set up a condition that you select only experimental papers, or only experimental evidence that is based on the papers. There are more questions.
I think the other question was addressed as well.
They are related.
Okay, someone is asking, do Go terms have evidence tags as well? Yes, there's more information actually on. So if you go into one of our protein pages, you have the list of the Go terms, and near that list you see a link to QuickGo, and this is an external website that has more detailed information. If you click on it, you can see all the complete list of Go terms, because in our UniRule entry we just select a few Go terms. But if you go to QuickGo by clicking that link, you not only see all the Go terms but the origin. So you also have evidence codes attached to it. So you can see, oh, this Go term is coming from this specific ARBA rule, or from this specific UniRule rule. I think we have another question. Does UniProt associate obscure protein sequences reported on literature, such as toxins from a prokaryotic genus? I'm not sure what you mean by obscure proteins. So what we do at the moment, so UniProt is it has some years, the things that we used to do like 20 years ago, it's different like the things we do now. But at the moment, what we do is we don't import protein by protein. We import proteomes. So the information that we get, they come from a genome assembly. And if that genome assembly has the translated proteins associated with it, we are going to import. I'm not sure if this answers your question, but this is to say that if this obscure protein exists in the proteome, in the genome assembly, then we are going to import. You can further explain to me what if you have an additional question on this, because I'm not sure if I addressed this. And then we have a user that says, I have analyzed RNA-seq. So maybe I'll read the question out loud. Hello, thank you for the webinar. I have analyzed RNA-seq data and attempted and translated alignment against the SwissProt database, but the annotation was difficult to get. Regarding Unifier, what constitutes the input data? Additionally, is it possible to cluster proteins into pathways if they originate from the same pathway? For instance, having two enzymes from the same pathway. So regarding your first question, which is Unifier, so you just need a FASTA file where you have the protein sequences and in the FASTA format. So you need to have a header, and the header needs to be in the UniProt format. So you need to have a description and also a taxonomic ID. So this is the minimal input you need for Unifier. And your second question is if you can cluster proteins by pathways. I'm not the right person to ask that. Now, do you know?
Yes. So the thing is that for some proteins, the specific pathways are assigned, but it's not. UniProt is not focused on the reconstruction of the pathways. So it means that although some of the proteins can be identified and described as participating in the certain pathway, it doesn't mean that all the proteins from these pathways will be described. So basically, you still can find the information about the pathways, but it doesn't mean that the entire pathway will be there. So some of the proteins might be missing if they were not annotated in the project.
Okay, I think maybe as that was our last question, we could leave it there, and to say again that if anyone would like to add questions with more detail to ask the wider UniProt team, of course they can use the help desk to do so, or indeed if you think of some questions later on after the webinar has finished.